Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-level biology topics. This is A2 Biology, and the first topic is photosynthesis, which is topic 5A. Here I begin with topic 5A1, which is the importance of ATP. We're going to be focusing on the ATP molecule and its use in metabolism. We begin by looking at ATP as a source of energy. Here the objective is about phosphorylation, where ADP is converted into ATP by addition of an inorganic phosphate onto ADP. But before we go further, I want us to introduce different categorizations of organisms in terms of autotrophs as well as heterotrophs. Autotrophs are organisms that make their organic compounds of food directly from carbon dioxide, which is found in the air, or it could be found in the water, wherever, but provided it's a form of CO2. Heterotrophs are organisms that eat plants or eat other animals or other organisms that have eaten plants. So humans and many animals we know are all categorized as heterotrophs, while plants are categorized as autotrophs. In the next part, I want us to talk about hydrolysis and condensation reactions surrounding ATP. So the breakdown of ATP involves addition of water, so this is going to be a hydrolysis reaction, and it's going to be catalyzed by the enzyme ATPase, making ATP go through condensation reactions, and these are also catalyzed by the same enzyme ATPase. For these reactions surrounding ATP, we have reactions we call catabolic reactions, and some are going to be anabolic reactions, and these two reactions are part of metabolism. Metabolism can be categorized as partly anabolism, or it can be catabolism. Catabolism involves the breakdown of certain molecules, while anabolism involves the making of certain molecules. So ATP synthesis is going to occur from anabolic processes like respiration, as well as removal of hydrogen atoms from intermediates in the metabolic pathway, while ATP usage is usually from anabolic reactions. So down here we see the demonstration of this is an ATP molecule. You can see there are three phosphates. There's a ribose and adenine. So these make the ATP, which we call adenosine triphosphate. When water is added, this is hydrolysis. So the forward reaction is going to be hydrolysis, catalyzed by the enzyme ATPase, and in the product side we form ADP as well as the inorganic phosphate, which is PI. That is for the forward reaction. However, if we are combining them, then it means a water molecule is going to be lost as this phosphate binds onto this phosphate. That means we'll produce ATP as well as water, so that will be a condensation reaction. The electron transport chain, we have molecules that are arranged at different energy levels, and these molecules are going to gain electrons and lose them through series of redox reactions. For example, when an electron is excited, maybe from a chlorophyll molecule, it's going to be gained by the first molecule. If this molecule gains the electron, we say it has been reduced. However, this electron is going to be lost to the next molecule in the chain, but this molecule is lower in energy, so as this electron goes down, energy is going to be released. So this molecule that has lost the electron, we'd say now it's been oxidized, as the one that is gaining that is going to be reduced. So again, this gets oxidized as it loses the electron to the next one, which is going to get reduced. So that is why I say there are a series of redox reactions. They involve reduction as well as oxidation, where reduction is gain of electrons and oxidation is loss of electrons to the next molecule within the chain. So this energy that has come off as electrons transition from a higher energy level to a lower energy level is going to be used in the processes that lead to formation of ATP. So I said here the electron transport chain is used by cells to get energy from the excited electrons, and then this energy is going to be used to bind ADP with PI in order to generate an ATP molecule. Then the components, of course, of this electron transport chain are going to go through reduction as well as oxidation in order to allow the electron to transition to the lower energy levels. Next, we look at topic 5A2. Here we're going to be focusing on chloroplasts as well as chlorophyll. Chloroplasts are organelles that contain chlorophyll. So here, of course, we can see the structure. We have the outer membrane. You can see on the outside there is an inner membrane. And then we have the stroma inside here. We have the thylakoids. These thylakoids contain the chlorophyll molecules inside them. We have the lumen inside there, we have the granum, which is the stalk of thylakoids, and so on. And chlorophyll is arranged within the thylakoids to maximize sunlight capture. So there is a specific arrangement that allows for maximum capturing of sunlight. We also need to know that chlorophyll is the one that traps the sunlight. This trapped sunlight is going to be used for the photolysis of water as well as the excitation of electrons from the chlorophyll molecule. I'm going to talk about this in detail a little bit later when we study about phosphorylation. In the thylakoids you see here, that is where chlorophyll is going to capture sunlight in order to produce ATP. And within the stroma, here ATP is going to be used to make glucose. You remember we'll talk about later the light-dependent and light-independent reactions. Those light-independent reactions that lead to the formation of glucose take place in the stroma, while the light-dependent reactions take place in the thylakoids as well as the lamellae. Before we go any further, we need to focus on chlorophyll. We need to know the different types of chlorophyll. There is chlorophyll A, which is blue-green. Of course, because it appears blue-green, it means it does not absorb well within that region. There is chlorophyll B, which is yellow-green. And then we have the carotenoids, which are orange carotene, as well as yellow xanthophyll. And then we have the pheophytin, which is going to be gray. So, and again, nobody's going to ask you to remember this from theory. Oh my God, what is chlorophyll A? What is chlorophyll B? Carotenoids and so on. But they will give you questions maybe in a graph. It could be like this absorption spectra here. The key thing we can get from this absorption spectra is we have many different chlorophyll molecules, and many of these molecules have different regions in which they absorb. The purpose of this is to ensure that maximum absorption of sunlight occurs. So if a plant has the varying chlorophyll pigments, it means this plant is going to be able to absorb sunlight no matter the light wavelength that is present in order to maximize photosynthesis so that it can make organic compounds. And still on the same topic, the absorption spectra describes the different amounts of sunlight of different wavelengths absorbed by different photosynthetic pigments, as you can see. Also, you need to know an action spectra demonstrates the rate of photosynthesis according to the wavelengths of light. If you can look in some of the past papers that I've answered previously, there was a question where they give absorption spectra and asked you to use that information in order to draw an action spectrum. So next we go to chromatography. Here we're learning chromatography because we want to focus on the idea of separating these chlorophyll pigments. Imagine you have successfully extracted some chlorophyll pigments out of a leaf or out of a plant, but you want to be able to know which molecules they are. You could use chromatography in order to separate them and then use the RF value in order to know which one exactly they are. Chromatography is used to separate mixture of components. In this case, you will need to extract pigments by grinding maybe with propanol. This is acting as a solvent in order to extract the pigments out of the leaf. Then the filtration process is done in order to remove the leaf residues and obtain the solution or the liquid that forms. Then you will carry out chromatography using the liquid that you obtain and calculate the RF value. Then you will use this RF value of the pigments and compare with information from the data booklet in the same solvent in order to identify the separated samples. Now, to carry out chromatography, you will need a chromatograph paper, like you can see this one here. Again, there is this that was given in your textbook, but here we can just see the different molecules that have been separated. I want to use this part here for my explanation. So you will have a chromatograph paper like that, and then you have to choose a suitable solvent. But before you put or dip this chromatograph paper into the solvent, you will have to use a pencil and draw a baseline. We use a pencil because the color or the lead is not going to interfere with the sample or with the colors that you're trying to visualize. So this is going to be a baseline drawn in pencil. And then you choose a specific solvent and put the solvent maybe inside a beaker. But you have to make sure the solvent does not touch the pencil line or the baseline. It just stops below the baseline, just touching the paper. Then you put the sample at this point. This is the mixture that you have extracted. Ensure that this paper is held somewhere so that it does not fall into the solvent. You have to find a way of anchoring it. Maybe you're clamping it or whatever. And then most times it's recommended that you cover because some of these solvents easily evaporate. They could be volatile. So if you cover, and then there is a way of anchoring the paper so that it doesn't fall through. So this solvent is going to move up the paper. And as it moves up, it's going to carry these components, separating them into different distances from the baseline. So you will wait for the solvent. As soon as it's coming closer to the end, mark the end point. We call this point the solvent front. Let the chromatograph paper to dry, and once it's dry, put it on a flat surface and use a ruler to measure the distance of the corresponding components from the pencil line, like that, and then also the distance of the solvent front from the pencil line. Once you know those distances, then you're going to use them to calculate the RF value. Now, using this as an example, the RF value of A is going to be the distance traveled by sample A divided by the distance traveled by the solvent, or the distance from the baseline to the solvent front. So you can see this distance is going to be X. That is why the denominator here is X, because the solvent has traveled from the baseline to the solvent front, which is this one here. That distance is going to be X. While the distance traveled by sample A, you can see is going to be distance A, which we see here. So A divided by X is going to be the RF value of A. And the RF value of B is going to be distance traveled by B, which is B, divided by the distance from the baseline to the solvent front. And that is what we get. So once you get this, you will use this RF value that has been calculated to compare with the values in the data booklet and see, or to be able to know, that this sample is exactly what they have in the data booklet. When you're comparing the results with the data booklet results, you have to make sure the solvent used in your experiment and the solvent quoted in the data booklet is exactly the same. The fact about chromatography is it deals with solubility. It depends on the ability of these components within the mixture dissolving in the solvent or interacting with the solvent as the solvent is carried along the chromatograph paper. So those that are interacting better are going to be carried a longer distance, and those that are interacting in a poor way are going to be carried a shorter distance from the baseline. Now moving to 5A3, here we're going to look at the biochemistry of photosynthesis. So here we're looking at photosynthesis. Photosynthesis is divided into the light-dependent stage as well as the light-independent stage. The light-independent stage occurs in the stroma, of course you can see here, while the light-dependent stage occurs in the thylakoids. Photosystems are complexes containing chlorophyll. It means within the chloroplast, we have the thylakoids, and then we have within the thylakoids, we have those photosystems where chlorophyll molecules are going to be contained. So these photosystems are categorized into two. There is photosystem 1 as well as photosystem 2, as we're going to see here. Now, remember during photosynthesis, chlorophyll traps sunlight. The purpose of sunlight is to cause the photolysis of water, meaning the splitting of a water molecule into a hydrogen ion, or in this case into 4 hydrogen ions and hydroxide ions. And again, if there are 4 hydrogens, it means 4 water molecules have been split. I used 4 here because I wanted to balance the electrons, otherwise it wouldn't make sense, because 1 water molecule is going to be split into 1 hydrogen ion and 1 hydroxide ion, but 1 hydroxide ion is going to be converted into electrons plus oxygen plus water. However, here we see we have to put a 2 here to make the oxygens even, and when we do, this is going to be a 4. However, the charge is not going to be balanced, and that means we have to put a 4 here to have 4 negatives on either side. When we do that, it means the hydroxide must have been 4, and therefore the hydrogens should have been 4, so the water should also have been 4. So balancing the electrons is what causes this to be 4 in the beginning. Now here to see the breakdown of photosynthesis again, remember we said we have the light-dependent reactions. These occur in the thylakoids. Thylakoids inside those, and these are light-independent that occur in the stroma. The light-dependent reactions need photosystems, or we say we have chlorophyll molecules that are going to be able to trap sunlight or absorb sunlight. However, these chlorophyll molecules are contained in PS1 or photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2 are part of the light-dependent reactions, and within these photosystems we have what we call the cyclic photophosphorylation and the non-cyclic photophosphorylation that I'm going to talk about later. ATP is going to be generated from the light-dependent reactions, and then there is another molecule which we call reduced NADP that is going to be generated, and these 2 molecules, I can call 1 and 2, are going to be sent into the next phase of photosynthesis, which we call the light-independent reactions, and in here they are passed through the Calvin cycle, and then CO2 is going to be fixed. This is fixation of CO2. CO2 is going to be fixed, ATP is going to be used, reduced NADP is going to be used, and then molecules like glucose are going to be made in the end. Now remember again, we said photosynthesis is divided into the light-dependent and the light-independent reactions. However, the light-dependent reactions are categorized into cyclic as well as non-cyclic photophosphorylation. For cyclic photophosphorylation, this involves only photosystem 1, while non-cyclic photophosphorylation involves both photosystem 1 and photosystem 2. So this is very important to remember that. Focusing on cyclic photophosphorylation, which occurs in only photosystem 1, here sunlight is going to strike a chlorophyll molecule, and when this occurs, electrons are going to be excited to a higher energy level, and these are going to be picked up by the first electron acceptor molecule within the electron transport chain. Let's take an example if it's this one here. When it accepts that electron, then the electron is going to be released to the next molecule in the chain, and in doing so, energy is going to be released. Then as an electron is released from the next to the other, energy is going to be released. Like I already explained, these are a series of reduction and oxidation reactions, whereby if a molecule receives an electron, it becomes reduced, and if it gives it away, it becomes oxidized. So this electron is going to transition until it returns back to the chlorophyll molecule. The reason as to why we call it cyclic is because the electron returns to the chlorophyll molecule, it does not leave. The only thing we get from cyclic photophosphorylation is that energy is released, and this energy can be used to form ATP. So here we can see energy for ATP production from the electron transport chain. That's all we can get from cyclic photophosphorylation. Now on the next page here, when we look at the second part of the light-dependent stage, which is the non-cyclic photophosphorylation, this is the second kind. This occurs between photosystem 1 and PS2 or photosystem 2. It occurs in both. In here, we will generate ATP as well as reduced NADP, unlike what occurred previously in cyclic photophosphorylation. In cyclic photophosphorylation, we only produce ATP. There is no reduced NADP produced. I'm going to begin with photosystem 1. Light is going to strike a chlorophyll molecule in photosystem 1, and an electron is going to be excited because when electrons gain energy, they vibrate. So in this case, these electrons vibrate until they leave the chlorophyll molecule completely. So this is going to be excited, and it goes through the electron transport chain, which are a series of molecules arranged at different energy levels. And as it does so, energy is going to be generated or it's going to be lost. These can be used to produce ATP. And then this electron is going to be picked up by NADP, or oxidized version of NADP. And then this electron, along with the hydrogens that came from the photolysis of water, are going to be picked up to produce NADPH or reduced NADP. There is a void here because one electron has completely left the system. Now to replace that electron, light strikes a chlorophyll molecule in photosystem 2. This leads to the release of an electron or electrons to a high energy level. These electrons can transition through the electron transport chain, and again here, energy is going to be released. This energy can be used in the generation of ATP, and this electron is going to transition until it's received by the chlorophyll molecule in photosystem 1. So you can see we call this non-cyclic because the electron never returns. This is different from what we saw here in cyclic. In cyclic, the electron left, went through the electron transport chain, ATP or energy for formation of ATP was generated, but the electron returned back to the chlorophyll molecule. But in non-cyclic, it never returns, it goes away completely. So this electron from photosystem 2 is coming to replace the electron that was taken away from photosystem 1 that never returned within the photosystem because it was used to form the reduced NADP. After this electron is gone, there is some electron missing here because we need to replace it. That is where these electrons that come from the photolysis of water come in, because they're going to come to replace the electrons that are being taken away from this first chlorophyll molecule in the photosystem 2. So in summary, an electron from the photolysis of water replaces an electron that has been removed from a chlorophyll molecule in photosystem 2. And then that electron that was removed from a chlorophyll molecule in photosystem 2 has gone on to replace an electron that was removed from chlorophyll molecule in photosystem 1. The excited electron from photosystem 1 has gone on and completely left photosystem 1 because it has been gained by NADP in order to produce NADPH. Now here, looking at the light-independent stage of photosynthesis, carbon dioxide is going to be fixed, and then the products from the light-dependent stage like ATP as well as reduced NADP are going to be used in order to produce compounds or molecules that are going to be used to make glucose. So this begins with a fixation. And in this process, Rubisco is the enzyme that participates in the fixation of CO2 by combining it with ROP, or what you call ribulose bisphosphate. When this occurs, remember this is a 5-carbon molecule while carbon dioxide has only 1 carbon. So when you combine these two, you form a molecule that is 6 carbons. However, this molecule has not been formed by scientists, but they assume it's there. It's a temporary molecule that could break apart to produce 2 3-carbon molecules, which are called glycerate 3-phosphate. You could call it just GP. And then this GP is going to be converted into GALP, or what we call glyceraldehyde 3-phosphate, by gaining hydrogen from reduced NADP, as well as conversion of ATP to ADP and PI as energy is released, and GALP is going to be formed. Again, here we have 2 GALP molecules, and these GALP molecules are going to be used later to form glucose. The remainder is going to be used to regenerate the 5-carbon molecule, which is ROP, that is later going to be used in the same cycle. This cycle is called the Calvin cycle. Now, questions are going to come about the idea of temperature and how temperature can affect the rate of photosynthesis. Remember, most of these reactions in the light-independent stage are enzyme-controlled, and therefore temperature is going to be a factor that affects the rate of photosynthesis, because the higher the temperature, the higher the rate of photosynthesis, or the higher the rate of enzyme-controlled reaction. However, if the temperature increases beyond the optimum, then the rate is going to go down, or the rate of photosynthesis or rate of enzyme-controlled reactions is going to go down. So in the exam, they could ask you questions about temperature and how it affects the rate of photosynthesis for the light-independent reactions. And for the light-dependent reactions, they'll ask you about sunlight and how it affects the rate of photosynthesis. Also, for the light-independent reactions, they could ask you about CO2 concentration. So this is the independent, and this is for the dependent. So looking at factors limiting photosynthesis, the first one is light intensity, and this is going to affect the light-dependent reactions. They depend on sunlight to cause the photolysis of water or to cause the excitation of electrons from chlorophyll molecules so that they can go through the electron transport chain, which later leads to the formation of ATP, as well as the formation of reduced NADP. Now for these two, these are mainly going to be in the light independent. So we have to think about carbon dioxide concentration. If it's going to be lower, then the rate at which CO2 is going to be fixed is going to be slower. And that means there is going to be less reactions going on. And this temperature is going to affect enzymes, like I've already explained. So here I put 2 graphs. In the first graph, you're studying the effect of CO2 concentration. If you see these 2 graphs, there is this one here and that one there. Both are going to be at the same temperature. But you see, as the concentration of CO2 increases, we see a higher rate of photosynthesis, even at the same light intensity. I'm going to draw a vertical line. Imagine this is the same light intensity. We have the same temperature, but the difference in CO2 concentration causes a difference in the rate of photosynthesis. We see at a higher CO2 concentration, there is a higher rate, and at a lower CO2 concentration, there is a lower rate. So for this graph here, CO2 concentration is limiting the rate of photosynthesis. Then when we go to this one here, we can see here temperature is going to be limiting the rate of photosynthesis. I'm going to use these 2 curves here to explain that. And again, I'm going to draw so that we use the same light intensity. So we're going to compare that value here and that value here. You can see the carbon dioxide concentration is exactly the same. The light intensity is exactly the same as the line I've drawn, but you can see they have different rates because they have different temperatures. So the higher the temperature, the higher the rate of photosynthesis. So here temperature is the limiting factor. So here temperature limits. So this brings us to the end of topic 5A, as well as to the end of this video. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.